Yes, certainly.
Okay. Well, I would like first to thank Maite for the nice introduction, and also the organizers of this webinar series of the European Calcium Society. I'm going to talk about Alzheimer's disease and calcium signaling and aging. And this topic will be also covered in the upcoming 16th International Meeting of the European Calcium Society in Cork, Ireland, with several important speakers like Ilya Vesprosvani, Estela Aria Gomez, and Alex Berkasti. Actually, Maria should be talking as well in this meeting, but she cannot make it, so that's why she was invited here as well. Aging is perhaps the most important risk factor for many neurodegenerative diseases, and also, of course, Alzheimer's disease. You can see here how the chances to get Alzheimer's of the number of cases increase exponentially with age. And this is particularly important in the case of Alzheimer's disease because it's the most important pandemia, one with about 6 million cases in the US right now, about 10 million people in Europe, and in Spain, about 1 million. 1/10 of the general population over 65 years old actually has Alzheimer's. So, and there is no cure for this disease, and probably the reason is because, and let me just show you the pointer best. Probably most people that has Alzheimer start the disease 20 years before the first symptoms. So this is one of the difficulties, the most important difficulty for achieving good results with clinical trials, even though the treatments are good, at least in animal models. The other important factor in Alzheimer's disease is known because of the familial forms of Alzheimer's disease, in which there are mutations in the amyloid precursor protein, or APP, or in the enzymes, the gamma secretase complex that cleaves this enzyme, containing, for example, presenilin 1 and presenilin 2. In these cases, or for a known reason, there is a increase in the forms of longer forms of A beta, containing 42 amino acids instead 38 or 40, and this longer form tend to aggregate more, leading to the formation of small soluble aggregates, or named oligomers, or large fibrils. All of them are toxic for neurons, but perhaps oligomers are 10 to 100 times more toxic than fibrils, particularly because they are not only neurotoxic, but also synaptotoxic, because they can accumulate in synaptic connections, leading to the damage of these connections. And some of these forms may lead to changes in the homeostasis of intercellular calcium. So there is an interplay between aging, Alzheimer's disease, and calcium, and this is going to be the issue we are going to be talking about. My idea is to briefly summarize our most important contributions in these years in 3 parts. First, the effects of amyloid oligomers on intercellular calcium, particularly on the role of mitochondrial calcium and the potential targets for these amyloid oligomers. The aging, the relation between aging and the susceptibility to neuron damage induced by different neurotoxins. And I will focus on the role of intercellular calcium in this case. And finally, the possible remodeling of intercellular calcium homeostasis in neurons aged in vitro, and focusing on the possible role of amyloid oligomers in this case. We started about, well, less than 15 years ago, investigating the effects of several forms of amyloid peptides, including fibrils, or a fragment, A beta 2535, that was known to be toxic for neurons. And we observed that these fragments induce large increases in intercellular calcium in cerebellar neurons, but fibrils were not doing that. We also reported that these oligomers, so they are probably dimers, trimers, and short forms of soluble oligomers, induce large and sustained increases in intracellular calcium in cerebellar neurons, as we did first, but also in hippocampal neurons and cortical neurons. You see here huge increases in mitochondrial calcium, similar to those recorded with activation of glutamate receptors by NMDA or depolarization of the cells using high potassium medium, which induce large increase in calcium. But our contribution in this area was the use for the first time of bioluminescence imaging to monitor the effects of these oligomers on mitochondrial calcium. And as you can see here, these neurons that are transfected with mitochondrial GFP equine, and this is a bioluminescent picture, display large increases in mitochondrial calcium, even to the millimolar level. So this is actually a mitochondrial calcium overload induced by the oligomers in these neurons. And we expected these increases to be really important. And to check that, we tried to prevent mitochondrial calcium uptake in these neurons and see what happens with survival. And we use over-the-counter drugs, including aspirin and several non-steroidal anti-inflammatory drugs, just because they are known to be mild mitochondrial uncouplers. And we use them because they were of interest in the field because they have anti-inflammatory activity. And it was supposed that Alzheimer's disease has a relationship with neuroinflammation. Even one of them, Fluthan, was used in a clinical trial, actually in a third phase clinical trial. And Fluthan is actually R-flubiprofen. So it's a non-steroidal anti-inflammatory drug, but they are an enantiomer that lacks anti-inflammatory activity. However, as you can see here, R-flubiprofen induces a loss of mitochondrial potential. This is the effects of FCCP on mitochondrial potential that actually collapses mitochondrial potential. And at very, very low concentrations of R-flubiprofen that has no anti-inflammatory activity, it's able to prevent largely, to inhibit largely the driving force for mitochondrial calcium uptake. So actually, this drug at this very low concentration of about or below 1 micromolar prevent mitochondrial calcium overload in these neurons and also prevent cytochrome C release. This is the cytochrome C release or apoptosis, which is essentially the same, induced by amyloid beta oligomers, and how it is largely prevented by this compound. Unfortunately, the clinical trial for this compound failed, but I believe, the field believes that this is probably because the disease has to be treated much earlier. In addition, we wanted to find the effects of, the direct target of these oligomers. And for this, we use a large array of chemicals and pharmacological inhibitors. For example, we use a blocker of NMDA channels, a blocker of PQ channels, a blocker of sodium voltage-dependent channels, and a blocker of amyloid channels. And we found that actually all of them prevented the effects of the oligomers. So how can be that possible? And we believe that this is because neurons in primary culture form a network with connections, actual connections, synaptic connections, and when one of the neurons is activated, then the neuron releases glutamate, activate glutamate receptors, then depolarization, calcium entry, and so on, so propagation of the action potentials around the network. So to test the effects of blockers, we need to work in conditions in which there are no synaptic connections, and we can work using TTX, or we can work early days of primary culture in which the connections have not been established yet. And in these conditions, we found that only the blocker of NMDA channels or the amyloid channels, NA7, are able to prevent these effects. And actually, we add them together, like in this case, we essentially abolish the effects of the oligomers. So we believe that amyloid oligomers induce calcium entry and mitochondrial calcium overload in neurons, probably acting as amyloid channels or activating NMDA receptors, and that several NSAIDs and, for example, R-fluorbuprofen prevent mitochondrial calcium overload and protect against these oligomers. So this is the influence of these oligomers on calcium. But we wanted to know the influence of aging in this susceptibility of the neuron to die by neurotoxins. So we use different, to introduce aging in this problem, we try to use a simple model. And we introduce the model of a long-term culture of rat hippocampal neurons in primary culture, because it was established by the group of Doty here in Spain that the long-term culture of these neurons in specific conditions led to several aging hallmarks like these ones. So we wanted to see the effects of several neurotoxins on young neurons, which are short-term culture hippocampal neurons, or age neurons, which are represented by long-term culture hippocampal neurons. And we tested the effects of NMDA, which represent an stimulus for excitotoxicity, amyloid beta oligomers, or LPS, which activates toll-like receptors and induce neuroinflammation in neurons. And treat them with 1 hour for 1 hour, and then wait for 24 hours before testing apoptosis. And we found that in general, in young neurons, the neurotoxins like NMDA induce significant toxicity, but this toxicity increased dramatically in aged neurons. And we got nearly the same results using amyloid beta oligomers, some toxicity in young neurons, but enhanced dramatically toxicity in aged neurons. And the same results were obtained essentially with LPS, lipopolysaccharide, acting as a pathogen-associated molecular pattern activating toll-like receptors. We wanted to check the effects of these neurotoxins on calcium, and as we expected, we observed that calcium rises induced by these toxins were enhanced dramatically in age cultures. Here you can see the effects of NMDA. Exactly the same results were obtained with the simulation with amyloid beta oligomers with 142, the longer form. And essentially the same result was obtained with LPS that does not induce increases in calcium in young neurons, but produces increases in calcium, dramatic increases in calcium also in aged neurons. We investigated the possible molecular basis of this and investigated using semi-quantitative immunofluorescence the expression of several subunits of the NMDA receptors, and the subunits NR1, NR2A, NR2B, and found that the first two subunits increased with age, in vitro age, but however, the third one actually decreased significantly with age. And these changes mimic those found in vivo. So they kind of suggest that the in vitro model of aging can be a good model. We also investigated the effects of aging, in vitro aging, on the expression of toll-like receptor 4, which is the target of LPS, and found that these receptors are also overexpressed in aging. And one important result is that if we treat the cells or the long-term cultures with amyloid oligomers in dark gray here, we observe that the expression of toll-like receptor 4, the target of LPS, also increases after treatment. So kind of mimicking the effects of aging in young neurons. We also are interested in other kinds of neurotoxins that became very important in the last few months, and are interested in SARS-CoV-2 virion because this virus has killed a lot of people. Many people has also central nervous system symptoms. They infect neurons, promote headache, memory loss, impaired consciousness, and epileptic seizures. And we focus on one of the 30 proteins of this virus, which is the envelope protein, which is critical for propagation of for the replication of the virus. And we pay attention to this protein because it actually is a viroporin. It's a integral membrane protein with a single helix that can form ion channels. We observed that actually just incubating the neurons of the neural cultures with this protein is able to induce apoptosis, but only in aged neurons, indicating that, again, our model allows to investigate the effects of increased susceptibility to neurotoxins. We are also investigating the effects of this envelope protein on calcium, and found that there are this protein, which is a 75 amino acid protein, so about 2 times the size of the amyloid peptide, increases intracellular calcium. Probably the effects is larger until, but we cannot be absolutely sure right now. These effects are mediated by release of calcium from intracellular stores. So we can conclude from this part that in vitro aging enhances calcium responses to neurotoxins and the susceptibility to damage. And finally, I'm going to talk briefly about the possible remodeling of intracellular calcium homeostasis in neurons aged in vitro and the particular role of amyloid oligomers. And we use the same model of in vitro aging, and we studied different parameters related with intracellular calcium homeostasis. We started by measuring the calcium store content by the calcium rise induced by ionomycin in the absence of extracellular calcium. And as you can see here, in aged neurons, the calcium rises are larger than in young neurons, indicated the calcium store content in aged neurons is larger. And we treated both young and aged neurons with amyloid beta peptide oligomers and found that there was no effect on the size of calcium stores in young neurons. However, in aged neurons, treatment with amyloid beta oligomers further increased the calcium store content. So here is the data. Amyloid beta oligomers has nothing in young neurons, but it increases further the calcium store content in aged neurons. Similar results were obtained when we measure calcium release using acetylcholine in the absence of extracellular calcium. You can see here the calcium rises here, probably not larger, but affect more neurons than in the aged neurons and in young neurons. Treatment of neurons with oligomers has no effect in young neurons, but induces a dramatic change in aged neurons. So again, treatment with oligomers induces an increase in the calcium release induced by acetylcholine. So it kind of exacerbates the effects of aging. We wanted also to know what happens with the transfer of calcium from the endoplasmic reticulum to mitochondria. And for this, we measure mitochondrial calcium rises induced by calcium release evoked by this agonist. You can see here that again, that mitochondrial calcium transfer is large in aged neurons compared to young neurons. And when we treated these neurons with amyloid beta oligomers, we found there was a dramatic change in treated neurons. And so amyloid beta oligomer treatment enhanced the calcium transfer from ER to mitochondria in young neurons. And this is the only effect we have seen in young neurons. And we know that this cannot be due to differences in calcium store content or calcium release, because I just saw that there was no change. So when we treated age neurons with amyloid beta oligomers, we expected to have a much larger increase, but instead we observed that actually calcium transfer between the ER and the mitochondria were kind of collapsed. We still don't know why. Finally, we tested the effects well, this is the average result. So in young neurons, treatment of amyloid oligomers increased calcium transfer from ER to mitochondria, and in young neurons, it does the opposite. And finally, as I said, we tested the store-operated calcium entry, which is a critically important calcium entry pathway in neurons, because it allows memory storage, because it affects the stability of muscle spines. And as you can see here, and that's been reported by other authors, calcium store content is decreased in aged neurons. And treatment with amyloid beta oligomers has no effect, again, in young neurons, but decreases it further in aged neurons. So we conclude that in vitro aging induces calcium remodeling in neurons, and this effect is kind of exacerbated by the presence of amyloid oligomers. We are, of course, trying to learn, and next speaker will talk about these things probably, if these changes or these differences actually occur in vivo. And another thing is to investigate the molecular basis of these changes. And for this, we are now investigating using transcriptomic analysis of calcium remodeling in vitro agent ratipocampal neurons. Of course, this is very long to tell now, but just as a clue, just show you, I would like to show you that how mitochondrial calcium transport is deeply affected by aging in ratipocampal neurons. Just to finish, I would like to mention this is the group of intracellular calcium pathophysiology, but most of this work has been directed by Professor Lucia Núñez and has been actually carried out by former PhD students in our lab, Dr. Sara Sanz Blasco, Erika Caballero, and María Calvo-Rodríguez, who is author of most of the work presented here. Thank you very much.
Thank you, Carlos. Very nice talk. And now it's the turn of María Calvo, who is going to talk about the in vivo imaging of mitochondrial dysregulation in a mouse model of Alzheimer's disease. Please, María.
Thank you, Teresa. Let me share. I got frozen, I think. Okay. I cannot share.
And do you maybe want to try the entire screen again? Yeah, we can see now.
Okay, perfect. So can you see full mode now?
Yes.
Yes.
Okay, perfect. Thank you. Thank you, Teresa, for the introduction and also for the organizers for the opportunity to present this work here. So as Carlos said, after we study those projects in his lab, then I was really interested to see this, but what it makes sense for me was to study this in the next step that was in vivo. So what I'm going to show you today is this study that I carried during my postdoc that is about in vivo imaging of mitochondrial dysregulation in a mouse model of Alzheimer's disease. So here I'm showing you an electron microscopy image of mitochondria in normal conditions. We can see that mitochondria has an outer membrane, an inner membrane, there is cristae and the matrix, and it's involved in ATP production, calcium homeostasis that we just saw, in generation of free radicals, and also in apoptosis. And here I'm showing you the same image of mitochondria, but in Alzheimer's disease. And we can already see that it has morphological alterations. We can see that mitochondria are swollen, they are fragmented, and also the cristae are fragmented or even lost. And there is also a decrease of mitochondrial number. And it's been shown mitochondria are extensively impaired in Alzheimer's disease. There is a decrease in ATP production and a decrease in the respiratory rate. There is an increase in apoptosis. Mitochondrial membrane potential is decreased. There is abnormal mitochondrial dynamics, loss of mitochondrial biogenesis, and it's been even shown that A beta can accumulate in mitochondria. And if we take a look at the amyloid cascade hypothesis, so this is one of the hypotheses for Alzheimer's disease, that it goes from mutations in amyloid to dementia. So if we take a look at it, we can see that downstream the effect of amyloid, there is impaired calcium homeostasis and also oxidative injury. So the question here is whether there is mitochondrial dysfunction also in vivo. And previous studies have shown in vivo that there is cytosolic calcium dysregulation, there is cytosolic increase of oxidative stress, and also that mitochondrial shape and memory potential are impaired. These are all related to amyloid plaque deposition. So our questions today is to know, to see if mitochondrial calcium and mitochondrial oxidative stress are upregulated in Alzheimer's disease. So the hypothesis is that A beta aggregates lead to mitochondrial calcium dysregulation and also to oxidative stress, and this could lead to neuronal cell death in Alzheimer's disease. So for this study, I moved to the lab of Brian Baskey. He's really known for doing multiphoton microscopy. So multiphoton is the simultaneous absorption of 2 infrared photons by a fluorophore. This has less phototoxicity and less photobleaching, is less invasive. And the most powerful characteristic of multiphoton microscopy is that we can image the complexity of the CD brain, and we can also do longitudinal image. So we can image the same field of view over time, like few hours later, next day, following week, or even for months. So to do that, this is what we do. We remove a piece of skull, like this window, and this is called a cranial window. We remove a piece of skull and we put a cover slip, and then we glue the cover slip, and then we can image through the cover slip. And the mice are, let's say, happy for even months with the cranial window. So let me see if I can play the video. So this is an example of what we see with 2 photon. This is a volume. This is a C stack. It's like a confocal image where we can go. Let me pause it for a minute. Here. we can see from top to bottom this volume. So what I'm showing here, I'm gonna show you like over this presentation, is this type of volumes that we can image. And usually we can observe the virus or the AAV in green. And then we will see the vessels, that is what we follow to create a fluorescent angiogram. It's like a map that we can track over time, and that's how we identify where we are in the brain. And those are labeled with a dextran. And then usually we can see amyloid plaques that are, this example here is in blue. This is labeled with a dye. So those are the three characteristics that we can see usually with 2-photon. In this case, this is an AAV target to mitochondria in neurons. So those, we could see the neurites and we could see the amyloid plaque as well. Our first question was if plaque deposition can lead to mitochondrial calcium dysregulation in an Alzheimer's model of in a mouse model of Alzheimer's disease. So we use the APPPS1 transgenic mouse model. This is a model of cerebral beta amyloidosis. It has 2 mutations, and with that, this mouse deposits plaques around 5 months of age. To measure calcium in mitochondria, we use this calcium reporter. We use yellow chameleon, and we introduce the mitochondrial targeted sequence. So we made yellow chameleon go into mitochondria, and we use the human synapsin promoter to make sure that it goes to neurons. So as you may know, yellow chameleon is based on FRET. So when it binds calcium, the ratio between YFP and CFP increases. So an increasing ratio is an increase in calcium. So we first validated this reported in vitro and ex vivo, making sure that it goes to mitochondria, and it goes to mitochondria in neurons. And we can see that it colocalizes with this marker of neurons, NUN, but it doesn't colocalize for a marker with astrocytes, for instance. And we also validated it in vivo to make sure it's functional. The way we did this, we injected a naive mouse, not transgenic, with the reporter, and then we implanted a window, and topically we apply KCl to the polarized cells. And we can see this field of view, how the calcium is increasing over time when we apply KCl. So we could see how it went from blue colors, so low calcium, to red colors. High calcium. And this is expressed, this is also in this graph here. So now that we know that our reporter was working, then we injected it in our transgenic mouse model in the APPPS1 mouse. And first we studied post-plaque, once the amyloid plaques have been deposited in the brain. So the way we do this, we inject and implant a cranial window, and then it needs some time for the brain to heal and for the virus to express. So we give it like 3 weeks. We inject methoxy to observe plaques, and then 24 hours later, we image. And this is what we observe. So what I'm showing you in these images, this is similar to the video I saw before, where we can see our neurons in green, and those are in red, those are the vessels. And on the right, those are the pseudocolor images. On the top is the wild-type mouse, and on the bottom is the transgenic mice. And we can observe how, for transgenic mice, we see those neurites already expressing high calcium. Whereas the wild-type mouse, they have low calcium in the neurites. This was also observed in somas, and we can compare those cell bodies in the wild-type and also the transgenic mouse. The way we quantify these volumes, to do that, we use MATLAB. So we have our C-stack, our volume, with these raw images where we see a lot of mitochondria, neurites, cell bodies. So using MATLAB, we use this adaptive thresholding that automatically MATLAB will do these ROIs for us, and it will surround every more or less mitochondria in that volume. So at the end, we end up with these histograms with thousands of mitochondria in that field of view, and then we have several randomized field of view per animal. So we can see how the histograms are shifted for the transgenic mouse to the right, meaning that calcium is higher in mitochondria. This is shown here in red. And we also define what we call calcium overload in mitochondria. We define calcium overload as the value for wild-type mice plus 2 times the standard deviation. So every number that would be like over this threshold here would we consider calcium overload. So this is in this the edge of the histogram, and this is shown better here. So we can see how calcium overload is higher for the transgenic mice compared to the wild type. So this is after plaque deposition. When we image the mice before plaque deposition, so young mice, we can see that we don't see this difference. So the histograms overlap for wild type and transgenic mice, and there is no calcium overload. Our next question was to know if plaque deposition can also lead to oxidative stress in mitochondria in this mouse model. So there could be mitochondrial dysfunction, then alteration of electric transport chain enzymes, which could lead to ROS. So to study that, we use a different reporter, and this one is a reporter that is for oxidative stress, and it's called ROIFP, in this case mitochondrial or mito-ROIFP. And we also install, we introduce the H-SYN promoter to make sure it goes to neurons. This is also a ratiometric reporter, but in this case, the is ratiometric in the excitation. So for that, we have to excite our fluorophores with 2 different wavelengths. So in this case, it's 800 and 900. So a ratio between 800 and 800 will mean that there is an increase in oxidative stress. As we did before, we validated it in vitro and ex vivo to make sure that it only goes to mitochondria, that we colocalize with another mitochondrial reporter, and also ex vivo, making sure that it co-labels with neurons, but not with astrocytes. And we also make sure that it was functional. So in this case, what we did, we image our field of view in baseline conditions, and this is shown in here. And then we apply an oxidizing agent. In this case, is DTDP. And we image the same field of view. And those are pseudocolors imaging. And we can use the same MATLAB script to create the same type of histogram. So we can observe when we apply our oxidative agent, so DTDP, it shifted the histogram to the right, meaning that there is an increase in ratio and therefore an increase in oxidative stress in mitochondria. So now that we know that our reporter was working, then we went to our transgenic mouse post-plaque, and we did it the same way as we did for calcium. So we inject some mice transgenic and also wild type, allow some time for the AAV and the window to heal and the AAV to express, and then we image sometime later. So in this case, I'm showing here the AAV, so mito-ROIFP in neurons. So those are some neurites, and there's a cell body there. And in this case, we are labeling the plaques with another dye that is called HS169, and it's in red color. So the good thing of this is that it doesn't leak into the green channel. So every signal that is in the pseudo color comes from only the AAV that is in the red channel. So when we quantify this, we observe that the overall ratio 800-900, so the oxidative stress in mitochondria was increased for the transgenic mice, as we can see in this graph. And also we observe how the histogram is shifted to the right for the transgenic animal. We also measure for young mice. And we can see how we have the same values for oxidative stress in the wild type and in the transgenic mice. Our next question, and that's where Carlos was showing before, is that if A beta oligomers, not only plaques, so also oligomers, so soluble A beta, can contribute to this mitochondrial dysregulation in vivo. So to do that, we use conditioned media. This is media that we collect from transgenic neurons that are in vitro and that are in vitro for 14 days, and then we collect that conditioned media. And we prefer using this media because it's more, the concentration is more similar to what we will observe in Alzheimer's patients. So in this case, we had 3 conditions. We have wild-type conditioned media. We have transgenic conditioned media that actually contains the soluble A-beta oligomers. And as a control, we immunodepleted the transgenic conditioned media with an anti-A-beta antibody. So the way we did this is is similar, but a bit different to what we did before. So first, we wondered if there was an increase in mitochondrial calcium that is produced by the soluble A-beta. So we use naive mice, so C57 mice, that were injected with our calcium reporter, the yellow chameleon, targeted to mitochondria. And the day of the experiment, we have the window implanted, and then we image in baseline conditions. And then what we did is that we reopened that window, applied the conditioned media for 1 hour, and then we image in the exact same field of view that we acquire of baseline images. So this is shown here. I'm showing the wild type conditioned media, transgenic conditioned media, and the control, the A-beta depleted conditioned media. So we can see that there is a before image, and then this is the after image, but in the exact field of views. So we can observe that only when we apply transgenic conditioned media to the brain, we can observe an increase in the calcium in the mitochondria. And this is shown here, how we can see that in baseline conditions, we see low calcium levels in mitochondria, but after application of conditioned media for wild type, there is similar calcium levels, same for the A-beta depleted transgenic conditioned media, but we can observe here how these red dots, so those are the mitochondria with high calcium. And this was quantified, so this is the delta of the ratio. We can observe how this is quantified here for transgenic conditioned media is significantly different from the other two. We could also evaluate oxidative stress following the same procedure. So to understand the contribution of soluble A-beta to oxidative stress in mitochondria, where we image before and then 1 hour after conditioned media. So we could observe how only application of transgenic conditioned media increase the oxidative stress in the mitochondria. And this is quantified here on the right. We also wonder if MCU is required for this mitochondrial calcium increase that is driven by A-beta. So to do that, we follow this same procedure I just showed you. But what we did was to use ruthenium-360 to block the pore to see if this calcium could enter into mitochondria after incubation with soluble A-beta. And we could observe that if we co-incubate the transgenic conditioned media together with ruthenium, the increase in calcium in the mitochondria is prevented. So I'm showing here the same images as before, where we can see before and after the same field of view. And we can see that with transgenic conditioned media, there's this increase in calcium, and application of both, this increase is prevented. And ruthenium alone didn't increase calcium. And this is quantified here. And we could also observe and wonder if blocking the channel with ruthenium could prevent the increase in oxidative stress in the mitochondria. And we could see how there is also less oxidative stress when we apply ruthenium together with transgenic condition media. And this is shown in these images and also in this quantification. Our last question was if we can see if chronic mitochondrial calcium overload can lead to cell death. So to do that, we use the same transgenic mouse, the one that was depositing plaques, and we follow different cells over time, so in 24 hours. So we can see when we image cells that have low calcium in the mitochondria. So this is yellow chameleon. This is the same reporter as we used before and the same transgenic mouse. When we have low calcium in the cell body, usually these cells remain over time. But when we follow cells with high calcium in the mitochondria, we observe how these cells disappear over time, and we assume they die. And we can also see this, how the nuclei get damaged. I have to say that this mouse model has very low cell death. So this is a really rare event. So it was not that easy to, like, we had to follow many cells to find this, but it was really surprising. I mean, we suggested that high calcium could also lead to cell death in vivo. So the take-home message of these studies is that both mito-yellow chameleon and mito-RFP can be expressed in mitochondria of neurons in vivo and report changes in mitochondrial calcium on oxidative stress, respectively. Mitochondrial dysfunction is increased in neurons in the APPPS1 mouse after plaque deposition as compared to before. A-beta oligomers, soluble A-beta, increase mitochondrial dysfunction in neurons when it is applied to the living healthy brain. MCU is required for the mitochondrial calcium increase and the subsequent mitochondrial oxidative stress increase induced by soluble A-beta. And we also think that mitochondrial calcium overload is directly linked to neuronal cell death. And the use of these tools could enable testing different therapeutics that are directed at reducing the mitochondrial calcium or mitochondrial oxidative stress associated to Alzheimer's disease in vivo. And with this, I, of course, want to thank Brian Baskey. He was a great mentor, and he helped me with all this project, and people in his group. Also people in the Hyman group, and also other people helping with MATLAB and statistics here in Mass General Hospital and Harvard Medical School. And this is what I had for today. Happy to take any questions now.
Thanks, Maria. Very nice talk. Great, both of you.